Every spring, colonies that survived winter in excellent condition swarm, not the weak ones, not the diseased ones, the strong ones, the ones with 6 and 7 frames of brood, with tight-capped honey arching above a deep winter cluster, with population building faster than the season can absorb it. That is not a coincidence. That is biology operating exactly as it was designed to. And yet, most beekeepers treat a swarmed colony as a management failure, as something that could have been prevented with more space, an earlier split, a younger queen. What those beekeepers are missing is that none of those interventions address the actual trigger. They address the symptom. The swarm was not caused by too many bees. The swarm was caused by what those bees perceived about their own hive. Have you seen this in your own colonies? A strong colony, brood in every frame of the lower box, the upper box packed with last season's capped honey, the bees building in numbers, consuming winter stores, preparing for a nectar flow that is still 6 weeks away. And then, before the first major bloom opens, half that colony is gone. If you've kept bees for more than 2 or 3 seasons, you have seen this. And if you are producing honey at any scale, 10 hives, 20 hives, 50 hives, that swarm cost you a significant portion of your main flow income. If this channel has helped you understand your colonies more clearly, consider subscribing. There is more to cover. What most beekeepers misunderstand is this. The swarm impulse is not triggered by population density alone. It is triggered by a specific perception the colony develops about its own storage capacity. And that perception can be manipulated without drugs, without splitting, without expensive equipment, by changing the arrangement of frames inside the hive. That is what this video is about. In the early 1990s, a beekeeper in Tennessee named Walt Wright began documenting a phenomenon he had observed across multiple seasons. Wright was not an academic. He was a working apiarist, managing a significant number of colonies across rural Tennessee, where spring nectar flows arrive quickly and compress into narrow windows. He could not afford to watch strong colonies swarm through the main flow. He needed to understand why they were doing it. What Wright documented, through direct observation and systematic frame manipulation, was that honeybee colonies develop swarm impulse not simply because they are large, but because they have filled the upper reaches of the hive with capped honey. When the band of capped honey above the brood nest becomes continuous, when there is no gap, no empty cell, no unsealed space that the colony can interpret as available storage, the workers transmit a signal through the cluster that the hive is full, not crowded, full. There is nowhere for new honey to go. The biological response to that signal is reproductive. Prepare a new queen, build swarm cells, and divide the population. The colony is not malfunctioning. The colony is responding rationally to what it perceives. Wright's insight was this. If you remove the continuous honey band, if you break that solid arc of capped wax with alternating empty drawn comb, the colony no longer reads the hive as full. It reads the hive as unfinished. And a colony that perceives unfinished storage work does not swarm. It forages. This practice became known as checkerboarding. To understand why checkerboarding works, you need to understand how a colony reads its own internal space. In late winter and early spring, as cluster temperature rises and brood rearing accelerates, the colony begins an outward expansion. The winter cluster has been tightly compact, consuming stored honey from the frames immediately surrounding it. As brood nest expansion begins, worker bees move upward and outward. They are not simply filling new space with honey. They are assessing available space and communicating that assessment to the broader colony through a mechanism researchers describe as overcrowding signals related to pheromonal communication and the spatial behavior of workers in the upper box. When a worker bee encounters a frame of fully capped honey, she has no work to perform there. She moves on. If every frame in the super or upper brood box presents the same condition, solid capped honey, no empty cell, No available surface. The cumulative signal that passes through the colony is one of storage completion. The hive has reached capacity. The colony's next reproductive step is triggered. By contrast, when a worker encounters an empty drawn comb or a frame that contains only partial storage, she has work to perform. She returns to foraging. She recruits. She stimulates other workers. The colony perceives itself as mid-task rather than task complete. That distinction, task complete versus mid-task, is the single biological lever that checkerboarding manipulates, and it must be applied before the colony reaches the task complete signal. Once swarm cells are capped, the decision has already been made inside the hive. No frame rearrangement reverses that. Checkerboarding is a preemptive intervention, not a rescue operation. The timing question is the most critical variable in this entire practice. In temperate climates, colonies begin serious brood nest expansion when external temperatures sustain above 10 degrees Celsius, roughly 50 degrees Fahrenheit, for consecutive days. This is not a single warm afternoon. This is consistent thermal stability that allows the cluster to relax and expand laterally and upward into previously unoccupied comb. In practical calendar terms, across most of Central and Northern Europe, and across much of the northern United States, this occurs somewhere between late February and early April, depending on elevation and microclimate. In warmer regions, the Mediterranean basin, the American South, parts of New Zealand, this window arrives considerably earlier. The beekeeper's task is to apply checkerboarding at the moment when the colony begins that upward expansion, but before the honey band above the brood nest has transmitted the full signal to the broader population. That window is typically between 3 and 6 weeks before your primary nectar flow begins. In most temperate systems, this aligns with the period before dandelion and fruit tree bloom, before the colony has reached peak foraging population, 3 to 6 weeks before primary bloom. That is the intervention target. If you apply checkerboarding during bloom, once the main flow has already opened, you have already lost a portion of the intervention's value. The colony may not swarm, but the foraging force that should have been accumulating honey during the early flow was instead managing swarm preparation internally. You cannot recover that time. The mistake happened weeks earlier. Here is what the physical intervention looks like. You are working above the brood nest. You are not rearranging brood frames. You are rearranging the honey storage frames in the super above, or in a 2-brood box system, the upper brood box if that box is primarily being used for honey storage at this time of year. You need drawn comb. This is a critical requirement. Foundation does not work. The colony needs empty cells that it can immediately begin filling. Foundation requires wax production, which requires warmth and population, and introduces a delay that undermines the entire mechanism. If you do not have sufficient drawn comb, begin building it now in your strongest colonies during any early flow. The pattern is alternating. 1 frame of capped honey, 1 frame of empty drawn comb, 1 frame of capped honey, 1 frame of empty drawn comb. Across the super, you are breaking the continuous honey band into segments, separated by unoccupied space. In a standard 10-frame Langstroth super, this means you are placing 5 frames of capped honey and 5 frames of empty drawn comb in alternating positions. In an 8-frame system, the same alternating principle applies. The empty frames should be full-depth drawn comb, ideally comb that has previously held honey and has been extracted and cleaned. Bees are drawn to familiar, used comb. They begin work on it quickly. After placing the checkerboarded super, close the hive. Do not manipulate further for 7 to 10 days. At your next inspection, 10 days after checkerboarding, you are looking for 3 things. First, are bees present in the new empty frames? Worker presence in the empty comb indicates the colony has accepted the task incomplete signal. Second, is there any evidence of swarm cell construction in the brood nest? If swarm cells are present and occupied, the intervention was applied too late. Third, Has the brood nest expanded into any adjacent space in the lower box? If bees are working the empty frames and no swarm cells are visible, the intervention is functioning. There is an important distinction between what checkerboarding prevents and what it enables. It does not prevent population growth. The colony continues to build. Brood production continues at its natural rate, governed by queen laying capacity and available cells. Population moves toward its spring peak on schedule. What changes is where the colony directs its energy once the foraging force reaches full strength. A colony that has not entered swarm preparation is a colony that continues to focus foraging energy outward, toward nectar, pollen, water, propolis. A colony in mid-swarm preparation is a colony that has turned partially inward, building queen cells, clustering around reproductive preparation, reducing foraging recruitment. The lost foraging recruitment during swarm preparation is difficult to quantify precisely, but field observations across multiple documented seasons suggest colonies in active swarm preparation may reduce effective forager deployment by 20 to 40% during the peak preparation phase. In a colony capable of fielding 12 to 15,000 foragers during a main flow, that is a substantial reduction in nectar intake. For a small producer managing 20 colonies, each swarming colony represents not just lost population, but lost production during what may be a 3 to 4 week flowering window. In regions where primary nectar comes from a single dominant species, linden, acacia, phacelia, clover, that window may compress further. Missing even 1 week of peak forager deployment from a swarming colony can reduce that hive's seasonal surplus by a material margin. Growth without timing is loss. The cost of a swarm is not the lost bees. The cost of a swarm is the lost days. There are two common errors in checkerboarding practice. The first is applying it too early in the season, when ambient temperatures are still below reliable brood nest expansion thresholds. If you checkerboard while night temperatures are still dropping to 4 or 5 degrees Celsius, 39 to 41 degrees Fahrenheit, the cluster remains compact. The empty frames are too far from the cluster warmth to be worked, and the intervention has no effect. The colony is not yet reading its upper box as full because it has not yet expanded into that space. You have simply introduced cold, empty frames into an otherwise insulated structure. Wait until the colony shows clear evidence of upward movement, frames of brood beginning to appear in the upper box, or worker traffic visible on the top bars of the upper super during mid-afternoon hours when ambient temperature is above 14 degrees Celsius, roughly 57 degrees Fahrenheit. The second error is checkerboarding with foundation instead of drawn comb. The importance of this cannot be overstated. Empty foundation presents the colony with a construction project, not a storage opportunity. The empty cells of drawn comb present an immediate task. Fill this. Foundation presents a prior task. Build this, then fill it. The colony's perception of available storage depends on the presence of actual empty cells, not potential cells. If your drawn comb supply is limited, prioritize it for your highest producing colonies. The colonies entering spring on 7 or 8 frames of brood, with the largest foraging populations and the greatest swarm risk. There is one additional timing layer that is frequently overlooked. Checkerboarding addresses the storage perception signal, but the colony also reads queen space. If the brood nest has become congested, if the queen has run out of available cells for egg laying, swarm impulse receives a second independent stimulus. Regardless of whether the honey band above has been broken, a colony with both a full honey band and a congested brood nest is under double swarm pressure. Checkerboarding alone, applied only above, may be insufficient in that case. The brood nest may also need direct management, removal of 1 or 2 honey frames that have migrated down into the brood box, replaced with empty drawn comb to restore laying space. This is not a complicated intervention. It is a 2-box inspection with clear diagnostic criteria. In the lower brood box, the queen should have at least 4 to 5 frames of empty cells available for laying at any given inspection during the buildup period. If capped honey has compressed the laying area to fewer than 4 frames, open the brood nest with empty drawn comb in the upper box. Apply the alternating checkerboard pattern before the honey band becomes continuous. Both interventions together address both independent swarm triggers. Either intervention applied alone addresses only one. The inspection sequence for a high-risk colony in early spring. A colony overwintered on 8 or more frames of bees should occur every 10 to 12 days, from the moment temperatures sustain above 10 degrees Celsius until primary bloom has opened and the nectar flow is confirmed active. That is typically a 6 to 8 week monitoring window. It is not extensive labor, but it requires consistency, more bees, less honey. The interval that matters is the gap between your last inspection and the moment the colony made its decision. Walt Wright documented his observations through direct correspondence with beekeeping researchers and journals during the 1990s, and his work circulated through hobbyist networks before receiving wider attention. He was not working within an institutional framework. He was applying systematic observation in a rural apiary under genuine production pressure. That origin matters. Not because of nostalgia, but because it reflects how a significant portion of practical apiary knowledge accumulates, not in laboratories, but in apiaries, across multiple seasons, by practitioners tracking outcomes against their own records. The biological principle Wright identified is not novel in a strict scientific sense. The role of spatial signals in regulating honeybee reproductive behavior has been examined in academic contexts. But the specific management application, the deliberate alternation of empty and filled frames above the brood nest as a preemptive swarm suppression strategy, was articulated clearly and practically through his field documentation. What Wright gave the production beekeeper was not a theory. He gave a specific frame count, a specific placement pattern, and a specific timing window. And he gave it without commercial interest. By the time the primary nectar flow opens, a colony that was checkerboarded at the correct moment looks different from a colony that was not. The checkerboarded colony has moved through its spring buildup without the metabolic detour of swarm preparation. It has not produced queen cells that were subsequently torn down. It has not reduced forager recruitment during mid-buildup. It has not lost 3 to 5 weeks of population growth to a swarm event that reduced its total forager count at the worst possible moment. It enters the main flow with its full foraging force intact, with empty drawn comb already positioned in the supers, and with a colony psychology oriented towards storage rather than reproduction. It fills supers, the colony that was not checkerboarded and that swarmed 2 weeks before main bloom, enters the same flow with half its bees, no swarm cells to requeen from, and a 6-week gap before its new laying cycle produces a functional foraging workforce again. That colony may produce minimal harvestable surplus during the main flow. It may produce none, reduces avoidable losses, stabilizes production, synchronizes brood and bloom. That is the language of field results, not promises. The bees have not changed. A colony in 1932 and a colony today respond to the same internal signals in the same sequence. The honey band above the brood nest meant the same thing to a working cluster in rural Tennessee as it means today in Central Europe or in the upland farms of New Zealand. What changes is the beekeeper's ability to read those signals correctly and to intervene at the correct moment. Checkerboarding is not a complicated practice. It requires drawn comb, correct timing, and a clear understanding of what the colony is reading when it reads its own interior. If you have strong colonies entering spring with full upper boxes, the decision of whether to act is already approaching. The intervention window is measured in weeks, not months, and the difference between acting within that window and acting after it is the difference between a colony that forages through the main flow and a colony that swarms before it opens. The mechanism is the same. The timing decides. The beekeeper adjusts.